Welcome everybody who are going to use BioLabDonkey program. In this video I will continue to present functions for analysis of DNA. And let's start with the function which can be used for generation of GenBank features database, and then we can use this database to find these GenBank features in our sequence. So let's start by pressing this button. When we press this button, we see this window. In this window, there is two sections, one for vector and one for genomes. Let's say we have several plasmids, vectors, and then we can make two different databases. If we would like to mix, doesn't matter, we can do this. Let's make vector database, the same applied for genomes. So first, we have to select the database where these features will be located. Then we should press this button. By pressing this button, we can open the files from which we can extract the GenBank features. Let's say we use this plasmid, and when we open this plasmid, then immediately all features from this plasmid are extracted and stored in local database. And then you see the description of this plasmid. And in this case, what I did, I just used the same plasmid as you see here. And what we can do, just to test this function, we can select this whole sequence, copy the sequence, and then we can open this sequence in the DNA vector tab, paste. Then we see this sequence. Just to remove all colors, we can use this button. We can clean the sequence in the main window, and as you can see in the graphic representation, there is no any features. And if you click on features window, then again we see no features. And let's say we would like to find GenBank features in this sequence. We already extracted the features from this plasmid database, and then we have marked this vector database, and then we can press this button, and by pressing this button we will find GenBank features in the sequence in the DNA vector tab. And as you can see, we found several features, and if we check if this is correct or not, we can switch to DNA insert tab. As you can see on the graphic representation, when we switch, we have absolutely the same graphic representation. So all features found in this sequence. By this way, I just wanted to demonstrate how we can use this function to make database for GenBank features and how we can search for GenBank features in our sequence. You also have option to export database as text file, and you can import database from text file which you generated before. One thing I have to mention, as you can see, we don't have in the window with GenBank features anything after we found GenBank features in our sequence. To see these features in this window, we have to press again this button to reload new features which we have found. As you can see, when I press this button, then these new features appear in this window. So this is all about this function. Let's close this. Next function is about plasmid map. When I press this button, we see the plasmid map, and also you see here the same features as on the graphic representation and in the main window. When we move on this arrow, then we see the description for this feature, and when we click on this arrow, we can immediately move in the main window to the sequence corresponding to this feature. Let's do it again. In this window, what you can do, you can remove or add plasmid name, you can remove or add features, description, and then you can change the position of features. Let's say it's a little bit far from the circle and then a little bit closer. If you have marked already restriction sites, let's do it, several of them. Then also what you can do, you can change the position of restriction sites in the same way as for features, just to have nice view for this plasmid map. Here you have option to change a little bit sectors for restriction sites. If I deactivate this function, then you don't see these lines. Sometimes it's convenient when the features located a little bit inside of the circle, then you see more clear which features are between these restriction sites. Then you of course have chance to change the color for the restriction sites. Let's see, green one, blue. If sometimes plasmid map has a lot of features and you just would like to concentrate on some features, you can double click on the feature. This feature will disappear on the plasmid map. If you would like to restore the initial view of plasmid map, you just press this button. This feature will appear again. If you see that the description for features are too long and not fitting to this frame, you can change the frame of this plasmid map, the size of this, and then you have a bigger frame for this plasmid map. And of course you also have the option for color for features, so you can change the color. So this is all about the plasmid map, and the next function is about text map. If you press this button, then you have this text representation of this sequence. In this case, it's more convenient because you see the two strand, forward strand and reverse strand, and you have option to see the several open reading frames. If let's say you would like to see the +1, +3, all frames, and also sometimes it's better to see only coding DNA sequence, and then you have to press this CDS. And then you see the protein sequence encoded by this DNA sequence. You can have at the end the list of features. If you sometimes don't need this list of features, you just remove this. If you have selected restriction sites, then you will see also these restriction sites here on this map. For example, this one. You can also print this, and this button is for the following function. If you, for example, have coding DNA sequence and you would like to use this sequence, you can select this part of sequence on one line, and then by pressing this button, you will immediately copy-paste this sequence in the protein tab. You see this sequence in the protein tab. If you select one more, and press again, then you will add more at the sequence which already in the protein tab. So this is all about text view map. Let's switch to the DNA insert tab. There is also a function to make virtual PCR. For this you have to press this button. To do this you have to first type primers. We can use the sequence as primers from here. We copy paste primer 1 and reverse primer for this one. What we can do, we select and then press this button, and in this window we copy paste the reverse complement primer and paste here. This is primer 2, and then select the sequence where we would like to find the PCR fragment when we're using these two primers. We also have option to use the whole sequence of the primer or 3' part of this primer. This is for the case when we have restriction site. In this case we can use the whole primer sequence, and then we have primers, we have selected sequence, and then we simply have to press this button. And by pressing this button we see the band which we'll have if we will run the DNA gel electrophoresis, and this is position for this PCR fragment. And if we select this line, then we have the sequence, and if we double click this sequence, then we should see this sequence in the main window. It's here. So this is all about the virtual PCR. Let's close this. And next, what I would like to show you is about the big sequences like genome DNA sequences. In this case, we have quite short one. It's only 3 kilobase around. But if we would like to open, let's say, let's close this, would like to open genomic DNA sequence, E. coli, we can do this. We can open this file. It takes a little bit time. to open, and as you can see in the main window we don't have sequence because it's too large, but we have the graphic representation from position 1 to 4 million base pairs around. And to see the sequence we have to zoom in. If we do like this, then we see the shorter sequence. In this case it's not short enough. If we know exactly which region we would like to use, then we can again select deeper and deeper, and when it's small enough, then we can see the features, descriptions, and also the DNA sequence. What we can do next, we can move on the left, on the right side, and as you can see, in this case, for this sequence in the main window, we have the restriction sites. If we would like to zoom in or zoom out, we can use these two buttons, plus and minus. And this is the zooming step for 3 kilobase. If we do like this, we zoom in, then we see the shorter and shorter sequence. If we would like to zoom out, we can use minus button. We can change the zoom in size. Default is 3 kilobase. And in the case of genome size, we don't have the option to see the plasmid because it's too large. It's bigger than 200 base pairs. But we can see the text map in this case, because for text map the sequence has the same length as in the main window, so it's around 21 kilobase. And we see the features for this piece of genomic DNA sequence. So at this point I would like to stop this video, and I will continue to present functions of this program in the next video. See you!